by Dr. Dhiraj Kumar from the International Center for Genetic Engineering and Biotechnology, New Delhi. And Dr. Kumar was elected fellow of our academy in this year, 2024. And the title of his talk is given there, Understanding and Solving an Ancient Disease Through Modern Approaches. Dr. Kumar.
I'm really thankful to the Academy for electing me and inviting me for the talk today. The title of my talk is Understanding and Solving an Ancient Disease Through Modern Approaches. So we have seen different flavors of scientific discussion in last 1.5 days. So I think this is one of the first where we will deal with infectious diseases, and the one that we focus on in our lab is tuberculosis. So ancient disease. So when we use ancient, we think of Egyptian pyramids and the Mohenjo-daro civilization. But tuberculosis is way older than that. The genus originated several 100 million years ago, and almost 3 million years ago, actually, the current tuberculosis strains kind of diverged. And the modern species of tuberculosis originated around 30,000 years ago. And while I showed just for the ancient reference for these Egyptian and Mohenjo-daro excavations and mummies in Egypt, they have been found to be having tuberculosis lesions when the X-ray and other analyses were done. So really, this is an ancient disease, and it has affected the mankind for a long, long time. And probably it's high time you start looking for solutions to this. So before the modern medicines became available, people were aware of what tuberculosis is and how it is impacting the life. There was no treatment at that time, of course, and the only solution was sanatoriums, usually at high altitude, cold places. The idea was that clean air, cold temperature is what delays the inevitable, but it hardly cured. Some of them were lucky enough to get cured, but nevertheless. And then in 1930s, when you had discovery of antibiotics, there was expectation that the era of bacterial infection will be gone because you have a really, the bullet to kill all the bacterial infections. But then it was a very short-lived euphoria because what I'm showing here is a chronology of some of the classic antibiotics discovery and when the resistance against those antibiotics were reported. So here is the year of discovery, and here it is the year of resistance identified. And you can appreciate that for something like carbapenems, the discovery of resistance happened almost in the same year when the drug was released in the market. So what I'm trying to emphasize here is that all antibiotics by design are destined to face this problem of drug resistance because we all know the Darwin's natural selection, and the antibiotics actually select against this very strong pressure that the bacteria undergoes when they are exposed to antibiotics. So tuberculosis is no different. Like all bacterial infection, TB also develops drug resistance. And this is almost 12 years old story, which came just around the World TB Day, which happens to be around 24th of March every year. Around 12 patients of totally drug-resistant TB was reported in Hinduja Hospital, Mumbai, and all of them died before their drug resistance profile was even measured at which drug they are susceptible to or vulnerable to. It's almost like road closed. Once you have totally drug-resistant, there is no way you can be cured. And given the antibiotics, vulnerability to developing new resistance, you need to come up with newer ways to target tuberculosis so that you can circumvent the problem of drug resistance. And so the work in my lab has mostly been focusing on those alternate approaches. So not really looking into antibiotics, but something which you can use to avoid the generation of drug resistance, and also something which works effectively in both drug-resistant and sensitive bacteria. And for that, we are targeting host mechanisms. Why host mechanisms? While I always use this slide to, kind of convey across the message that why host physiology is very important in driving TB pathology and how you respond to TB infections, almost all of us have witnessed in last 4 years what happened during COVID. So all of us responded very differently to coronavirus infection. And a very live example of that happened in a very unfortunate manner in Lübeck in Germany in 1930s, where in a neonatal ward, around 270 newborn kids were kind of inoculated with a virulent strain of mycobacteria instead of the vaccine strain. Vaccine was just discovered around that time, BCG vaccine. And all of those were inoculated with this virulent pathogen. Some of them died within 3 months of tuberculosis, around 70 odd kids died. And remaining 200 kids, they developed severe, moderate, mild to no symptom at all. Now, I mean, that was a disaster at that time, but now if you look back, this becomes a powerhouse information that how host physiology would have distinctly responded to the challenge they were exposed to, and kind of saved different individuals, but not worked in many others who were unfortunate and died. What it highlights that if you are able to understand host-MTB interactions in a much more clearer manner, probably you will have an additional handle where instead of antibiotics, you can modulate host functions, and that will allow you to control TB infection much more efficiently. May not be alone, you can combine it with antibiotics, but certainly it gives a new hope that you can develop something called host-directed therapy against bacterial pathogen. So it's an alternate way, and you can think of the added advantage that since you are targeting host and not the bacteria, the chances of developing drug resistance is minimalized because there is no natural selection pressure as such coming on the bacteria. Of course, it can come up with newer ways. How to identify which host factors, which host pathways are the main culprits behind allowing bacteria to establish, cause infection, cause disease, and things like that. They are certainly hidden inside us. And one of the earliest attempts that we did a long time back, this was my postdoc work early in 2010 and before that, where we did a genome-wide screen to identify are there host factors which specifically regulate intracellular bacterial survival, mycobacterial survival per se. And we identified several pathways and molecules which could be involved in regulating bacterial survival inside the macrophages. And once you identify that, you realize that almost all physiological processes together looks like they conspire together and allow bacteria to cause infection. It may be that bacteria is hijacking them in a way that it helps the bacteria more than helping the host. But just to give the highlight of the way interaction happens, so you generally inhale the bacteria which enters the lungs, and that's the most standard TB, but now we know that TB happens all over the body in different parts. And there in lungs, it goes to macrophages, which are the central and immune cells. And almost all the physiological processes that people study in innate immunity and in cell biology, almost all of them are perturbed upon infection. But over the time, for the long-term cellular responses, what happens that the gene expression pattern also gets perturbed, and that decides whether the bacteria is going to survive and grow, or it's going to persist or get killed, and accordingly, you will have active disease, you will eradicate TB, or you can have a latent infection which can then resurface at some other time when you are down in your immunity. So the 2 points, one is that host physiology plays an important role in tuberculosis pathogenesis, and if you understand it well, you can find ways to modulate, regulate it, and come up with host-directed interventions against tuberculosis. So my lab mostly focuses on all the host angle at different scales. So we look at tissue levels, where we look at different cell types which are recruited to the lesions where tuberculosis happens. This is called granuloma. It's this is human lung granuloma image generated from our group only in collaboration with AIMS, where we were looking at different immune cells which are coming to lung tuberculosis. And we looked for characterize different cell types, and we showed at how they can actually help bacteria to propagate better and cause infection. We look at intracellular trafficking events, that how bacteria goes from one compartment to another in a given cell. So we have looked at autophagosomes and how that interaction regulates inflammation, which is kind of hallmark during TB infection. We also look at different subcellular compartment trafficking, vesicle movement, bacterial uptake by the macrophages. What are the mechanisms that regulates the bacterial uptake? And at molecular level, we have been extensively analyzing gene regulation, splicing event, and whole genome variations, which can tell us whether you are susceptible, vulnerable, or tolerant to bacterial infection or not. So while I will not touch upon these probably some other time, so what I'm going to talk about in a little more detail in next few slides is the regulation of alternative splicing, which really has come up as an exciting discovery from our group, something which has never been reported in any bacterial infection, and that gave us some very interesting insights. So to begin with, we had done a simple microarray experiment. If those of you who have probably just heard about microarray, it just gives you the overall pattern of gene expression in a cell upon any kind of perturbation. You can give it heat, bacterial infection, or anything. And based on these heat color map, you can show the red ones are upregulated, green ones are downregulated. So it gives you the whole snapshot of the whole genome, what happens there. But that's microarray, which is like hybridization based. It doesn't give you a very clean picture. Why clean picture is required? Because eukaryotic genes are actually discontinuous in nature. When I say discontinuous, the coding regions are interrupted by non-coding regions, which are shown here as lines, and then they are stitched together in the process of transcription through a process called RNA splicing. So these are stitched together to form these functional messenger RNA, which forms the protein. Now these functional messenger RNA, you can appreciate here that these blue and gray boxes I have used exclusively in one case versus another. So what I'm trying to convey here is that these coding regions, or you can call them as exons, these are spliced differently to give different kinds of sequences, and you can appreciate that because of this, the protein sequence and structure also will be different because the coding regions are different. So from the same gene, you can have different, you can call it as splice variants or isoforms. That information you cannot get from normal microarray experiment, and any gene you pick up and put it on Ensemble, which is the standard database, you will find multiple splice variants of a given gene in the database. What we had done several years ago, we did RNA-seq analysis. It's a very high depth RNA sequencing analysis from the MTB-infected macrophages. And the observation was that infection and strain-specific perturbation of splicing happens in MTB-infected cells. And if you look at what are the functions which are getting affected through splicing, what you realize that these pathways, apoptosis, gene regulation, innate immune responses, these are the standard usual suspect pathways which will actually work against bacterial infection. All of them were getting targeted through alternative splicing. Then through functional studies, what we showed that if certain genes are expressed in a full-length variant, it will form a functional protein and lead to bacterial killing. But what happens during bacterial infection is that it gets expressed as a truncated variant because of alternative splicing, which I talked about, and then it is not forming a functional protein, and this helps bacteria to survive. Basically, without getting more dramatic, if this is how your host response looks like, this is what bacteria does. It breaks into pieces, and then the host response actually gets blunted. Now, this was a story published several years ago, and at that time there were questions coming from reviewers, what is the mechanism? Because splicing is a very complex process, involves assembly of large multimolecular complex, the number of steps involved, and all these circles shown, it actually is a RNA protein complexes, which recognizes which splice junctions to use, cut, and stitch together. So it's a very complex process. So how bacteria knows that how to regulate, which proteins, target mRNAs to regulate. And we decided to follow with a hypothesis that mycobacterial proteins can enter the cell and nucleus where splicing event generally happens. And when that happens, it can interfere with the recognition of these splice junctions, and that can lead to alternative splicing. It's a working hypothesis, and we went into that using different high throughput and candidate approaches, and this was supported thankfully by India Alliance, otherwise this work could not have been done. And we actually did the host pathogen interactome analysis. And without getting into detail, you can look at whether two proteins are interacting or not. There are very different kinds of methods. We use a special method called yeast-2 hybrid, where we had all the bacterial secretory proteins and human splicing-related proteins and just check whether they are interacting or not. And we generated an interaction map and looked like that a lot of splicing-related proteins were actually interacting with the bacterial secretory proteins. When that happens, then we did some in vitro assays to see that whether bacterial proteins can alter host RNA splicing or not. And you can kind of relate to these. These are the exons, and then the lines are the introns. So you can clone this whole thing and express these as a substrate, and then whether all 3 exons are retained or only 2 are retained, this is spliced out. You can do it by normal PCR reactions. And we did the in vitro splicing reactions, and you can see in this case 4 different MTB proteins were incubated during the reaction, and you see that the middle exon is retained in all the cases. But in case of another mRNA, the moment you bring the RV protein, this is the normal in the nuclear extract alone, but the moment you bring RV protein, the mesyl exon is spliced out. So what it means that in the presence of MTB protein, the recognition of mesyl exon is getting perturbed, and it is getting spliced out rather than getting retained. And this is something which is happening inside the cell, because in RNA-seq data, you are getting this differential regulation happening. So certainly MTB proteins can directly alter host RNA splicing. Now coming back to this, I was referring to these circles, that they are small RNA nuclear complexes. This is how it looks like. It's a very complex RNA, which shows these hairpin loop-like structures and interact with several proteins. And these are called snRNP, which has SMN complex and SM core rings. And what we found in the yeast 2 habitat, SMN complex proteins and SM core rings, both of those were interacting heavily with the MTB proteins. So we went back and did some more analysis, and what we realized that snRNP biogenesis is a evolutionary conserved process. It requires snRNA to be synthesized in the nucleus. It comes to the cytosol. It interacts with SMN complex, and then this heptameric core ring assembles over it. This is now snRNP. It goes back to the nucleus, where it participates in the splicing reaction. Now what our data suggests, that when this whole process is happening in cytosol of the cell, mycobacterial protein access them and they interact with these. And they interact and kind of sequester these in the cytosol. So once that happens, these proteins are not available to form these functional snRNP, and you will have a depletion of snRNPs in the cell, and that impacts overall splicing efficiency. So you have interaction of MTB proteins with these important complexes, and you have cytosolic access. You don't need bacterial protein to go into the nucleus. Even in the cytosol, they are able to access the splicing machinery, because the whole complex comes to the cytosol. It's not restricted only to the nucleus. And that's the interesting part which we discovered while doing this work, that it's interesting. And just to show the example, this is how the bacteria, so these are the bacteria which are labeled with certain fluorescence marker, and you can see this is one of the SM core protein called SNRPF, which nicely decorates the bacteria. And this is never shown in case of any bacteria, forget about MTB, that it can interact with the splicing machinery such beautifully. And whenever the splicing is studied in different contexts, in cancer, in spinal muscular neurotrophy, so in a lot of contexts splicing has been studied. In fact, the SMN complex I was referring to, the name is actually referred from spinal muscular neurotrophy, SMN. So what is known in those fields, that any alteration in the complex components leads to massive alteration in the pattern of splicing. And everything that we referred earlier, we were seeing interaction with SMN protein, interacting with SM core protein. Ideally, they should all lead to altered RNA splicing, and what we see during MTB infection is widespread splicing defect, several intron determination cases, and you get a lot of transcripts that do not get translated. So basically, that's the mechanism which in other contexts people have worked out. Looks like the exactly same thing is happening during Mycobacterium tuberculosis infection. And bacteria is smart enough to know that if you alter this snRNP biogenesis process, you have a control over the cellular response machinery. So while you express the gene, but it will not get into the functional form. And that's the beauty of the whole thing, that it happens. And then we did some more assays. We made some knockout of the mycobacterial protein we had identified and showed that it is involved in pathogenesis. And we also showed in mice as well as in human TB granuloma. So when you see human TB granuloma, there is strong staining for SNRPF, which is the protein we were looking at, suggesting that RNA splicing alteration is a very strong correlate of TB pathogenesis. And if you have ways to alter that, probably you will have newer ways of targeting tuberculosis. Now, splicing people are trying to target in cancer, but there is no report in TB, and that is something which we will try to develop in future. I'll just finish in 2 slides. to some extent, we have done some analysis with the interactions, surface between the host and bacterial protein in collaboration with the structural biologists who are easily available in ICGEB. And we are trying to develop that to disrupt those interactions and see whether that helps. Some early results looks good. Last slide. This process of nucleocytoplasmic settling is a evolutionary conserved process from yeast to higher eukaryotes, right? And this is a quality control pathway, because if snRNP biogenesis happens only inside nucleus, some immature snRNP can start participating in the splicing reaction. And that is very deleterious, because they will start recognizing even the non-genuine sites for RNA splicing to happen. So this is a quality control measure. What bacteria does, it hijacks this quality control pathway. And we looked at the bacterial protein that we identify that interacts with the spliceosome. In all virulent mycobacteria, its function is unknown. In all non-virulent mycobacteria, it is actually a beta-glycosidase, secreted beta-glycosidase, suggesting that for a non-pathogenic soil mycobacteria, it's actually just used as a different carbon source. It can grow a different carbon source. But probably when it acquired the virulence and pathogenicity, it acquired the ability to alter host RNA splicing. Now, it's too big a statement to make. I'll have to put the non-pathogenic bacterial gene into the pathogenic bacteria and show what happens there. But still, the way it looks like, it tells us probably the evolution of pathogenicity in the context of Mycobacterium tuberculosis. So I'll stop here. Summary is exactly what we have already discussed. I'll acknowledge the people in my lab, especially Komal, somewhere here, who was actually mostly it was her PhD work, and she has left. Funding from India Alliance, very well acknowledged. This could not have been done without their support, and of course collaborators and everyone else. Thank you so much, and we'll happy to answer any question.
Thank you, Dr. Kumar, for a nice talk. Now, can I take a few questions?
What actually triggered this tuberculosis strain to appear sometime 150 million years ago? That was the time, it was in geological report, it was Jurassic, when warm and humid conditions prevailed across the earth, and Indian subcontinent witnessed lots of transgressions.
Probably.
So what was its predecessor?
Those events happened probably when it was still, the evolution was happening in Africa, and because it's almost 100 million.
Evolution of what in Africa?
The pre-hominids.
So no, you talk 150 million years ago. That was
no.
Hominin.
No, that time nothing was there. But I'm saying that the pre-hominids, and they were getting infected with mycobacteria. Those events didn't happen that bang, but it started probably in Africa. And
150 million years to 1.7 or 1.5 million years is a huge time.
Yeah.
So
It's a huge time span. Yeah.
Appeared. The other thing I have a question. You said 5,000 years ago the strains are found in Egypt, and 3,300 years ago in the Indian subcontinent.
Yeah, those are. I wouldn't say with very strong conviction that years are exactly same. It can be 2,000 to 2,000 years.
So as a layman, I like to know whether the tuberculosis spreads during warm and humid conditions or dry and cold conditions.
Warm and humid. So
warm
you guess.
So that was the time when the conditions in the Indian subcontinent were cold and dry, 3,300 years ago, and 5,000 years ago also in Egypt.
Yeah, I have no answer for that. The only interesting part is, from these mummies, these guys were able to isolate bacterial genome, and they sequence it. And when you analyze that, it's almost 99% kind of retained even in the modern current day bacterial isolates. So much is the level of conservation in mycobacteria. So that's very unique aspect about it. I don't think for any bacteria you will have such long history of genome sequence available. So they actually went and did it from the Egyptian mummies isolated, and that was a Nature paper almost 10, 12 years back. Yeah.
During British time, people used to go to Rani Khet where lots of pine trees
So one of the images I showed, that is still there in Nainital. So that sanatorium is still there in Nainital. It's different matter, and nobody goes there because you have antibiotics and all.
So
one question, right at the back. Nice talk.
Thanks.
Is there any evidence to suggest that XDR strains would behave differently when they're splicing compared to susceptible strains? That's the first question. The second question is, when you said that this has not been described in other bacteria, is it because people haven't looked for whether this is happening in other bacteria that live in macrophages?
So for the second question, most likely people haven't looked at. There is one RNA-seq study done in Salmonella where you do see signature of it happening, but nobody has followed it seriously. Regarding the MDR strain, it can happen that some certain drug-resistant strain alter RNA splicing differently for simple reason that people have characterized secretome from normal wild type and MDR TB samples, and it is found that the secretome varies dramatically, right? So some of the proteins get heavily secreted in the MDR variant and not in the normal wild type sensitive strain, and vice versa. So if that happens and they have the, they are from the list that we have identified interacting, probably you will expect that some differences are there. But we haven't followed with the MDR TB cases as such.
Thank you.
Thanks.
So what is that SMN complex?
So SMN complex is a, there are a lot of these proteins and SMN proteins. The name comes from spinal muscular neurotrophy. So in Drosophila, people identified genes which were involved in, you can see, that mutation leads to SMN phenotype. So they named it as SMN, but, it recognizes the 5' end of the snRNA and binds to it. And once that binds, it's a step called assemblyosome formation that allows assembly of other proteins on the snRNA. So it's part of the snRNP biogenesis process, very well characterized in literature.
What is the main function of it?
No, so main function is allowing the assembly of snRNP to happen, right? So without that binding, your other heptamidic ring I was showing will not come to this. So it allows that recruitment process to happen, right?
The very strange splicing mechanism, wherever it is known, are those bacteria also equally ancient or they vary?
No. So splicing is happening in the host. So it's not happening in. So bacteria is only driving it in slightly different way than what happens usually, right? And that study that when a bacteria enters, the splicing may get affected, is not done in other bacterial cases. So that's what I was trying to. Yeah.
Thank you, Dr. Kumar.
Thanks.
Again. And